everyone. Welcome to Next Breakthrough. My name is Francesca Chen, and I have a surprise for everyone today in this month's episode. I interviewed Dr. Michael Gordon, a professor and PI in the Department of Zoology at UBC, whose team looks at a very unique animal in their research, the Drosophila melanogaster, a fruit fly. Intrigued? I was as well when I initially read through Dr. Gordon's research, and thus wanted to bring his thoughts to share with you all today. As the topic of this episode suggests, we will cover a wide variety and depth of aspects today, including fly models and genetics, neuroscience, sensory processing in terms of taste and scent, optogenetics, nutrient sensing, the connectome, and linking everything back to human health, and much more in much more detail. Although some things discussed do not directly relate to biomedicine per se, remember that biomedicine is really influenced and impacted by a broad scope of areas, and it can be through profound research from one of these areas that ties to something more central, as Dr. Gordon explains today. This episode gives us a broader view of the stretch and connectedness that are human biomedical sciences. It is all very interesting, so definitely go ahead and enjoy this unique episode. I would also recommend you guys research a bit about the Human Connectome project when you finish listening. So there's a bit of homework for everyone. Without further ado, here is Dr. Mike Gordon.
Hi,
Hi, it's nice to finally meet you.
you too.
So I think we'll just start by talking about the really unique animal that you use in your research. Forgive me if I pronounce wrong, but it's a fruit fly, right? So Drosophila melanogaster? Is that correct?
It's close. You're close. It's
All right.
Drosophila, Drosophila
Drosophila
melanogaster.
melanogaster. Okay, that was not close, but thank you very much. All right. Um, how did it become the go-to model organism for your research?
Well, it's probably surprising to a lot of people actually how much research there is in the world on Drosophila. So there are thousands of labs in the world studying fruit flies, and this one species of fruit flies. And so the question is, I guess there's two ways of looking at this question. One is like generally, how did the field end up studying this one animal? And then how did I actually end up in this field? So, to answer the first very briefly, a lot of it is sort of has to do with happenstance a long time ago. That somebody happened to observe this fly that had a mutation that caused its eyes to be white instead of red, and so he captured it and bred it, and that was sort of like the beginning of fly genetics. And it just gained so much momentum because they're really easy to grow in the lab. They are really easy to do genetic things with. So crossing them together, making, transgenic lines, knocking things out, all those things are really easy in flies compared to many species. And so then it caught hold in the beginning largely as a developmental biology model, where people made really important discoveries on like how genes and proteins regulate the process of development of an animal, and then expanded widely, including neuroscience and some particularly like well-funded groups became interested in using the fly model as model for neuroscience. So my own personal like journey into flies basically was when I was an undergraduate back in at McMaster University in Ontario. I just wanted to get into research, and I didn't really have a huge preference or understanding what I wanted to do. And the lab that I found space in was a fly lab, and I just sort of never left them.
Yeah. And now you have your own lab, which is really great, and you're still continuing with studying this fruit fly.
That's right.
And
Yeah.
to clarify, your research is on sensory processing and the behavior of these fruit flies. Could you maybe elaborate on that?
Sure. So We study mostly the taste system and how it relates to feeding behavior, but essentially we're interested in the question of like how an animal detects chemical signals in its environment and then how the brain of that animal processes those cues to elicit like robust and reliable behavior. As you might imagine, it's important for you, for any animal to make the right decision when it tastes, something sweet or something bitter or whatever, as to whether to eat it. And so, yeah, in general, we're just interested in how that decision gets made, all the steps from how the actual little protein receptors in the neurons in the fly detect those chemicals, all the way to how it gets translated into behavior.
Yeah, and what makes the fly's taste system such a unique tool for understanding taste, like in general, even in humans?
Um, so I think that it boils down to the same reasons why people use flies for anything.
Mhm.
Genetically, they're really tractable, and there's a lot of resources out there for studying them. And on top of that, amazingly, despite all of the evolutionary time between flies and humans, like the way they solve sensory processing problems has a lot of similarities, actually, in terms of how information is encoded and how it's processed, at least to the extent that we know. So they're a good model where we can just do things much more easily.
Yeah, and if I could bring in, I think you did a 2018 study on salt taste coding, if you can recall that paper, and
Sure.
it showed it was fundamentally different from other primary taste modalities. And can you
Yeah.
explain like what you mean by this and why it's so significant?
Yeah, for sure. Okay, so there's a lot of interest in neuroscience in how information is encoded. And what we mean by that is like if you have, let's say, a taste or a smell, what is the pattern of activity that that stimulus produces in neurons, either, in the periphery or in the brain of an animal that reflects the identity of that stimulus. So in theory, for example, like if I smell peanut butter, you could look in my brain and look at the activity and without knowing that I was smelling peanut butter, you could in theory figure it out based on the patterns of activation of neurons in my brain, right? Because presumably I have to be able to tell that it's peanut butter and not like bananas or whatever, right? And so that there's a lot of interest in understanding like what are those codes in neurons that that represent that particular stimulus. So in olfaction, which is, also a chemical sense, um, the bulk of what we know about it suggests that the that essentially animals can have very specific codes for all sorts of different stimuli, right? So estimates for how many different odors humans can discriminate between range from like, hundreds of thousands to a trillion or something like that. Like, but whatever the estimate is, it's a lot. Okay. So the brain is able to tell all these different smells apart. And the way it does that is to have different combinations of neurons active in response to those smells. Right. So you might only have, you have I guess a way of putting it is that like you can tell apart more smells than you have neurons that are actually encoding smells. And so it's all about the combinations of neurons. Okay. Whereas taste has always been thought of as being different. And tastes we think of as having dedicated populations of neurons for every modality. And there's a theoretical benefit and also downside to doing it this way, which is that, if you have dedicated populations of neurons for every modality, on the plus side, in theory, it's very easy for the brain to tell them apart, right? So like there's no ambiguity of like looking at all the huge population and thinking, okay, which specific combination is being encoded here? Like if a neuron that encodes sweet is active, then you're tasting something sweet. And if a neuron that's encoding bitter is active, you're tasting something bitter. And so by separating these two things, in theory, it's more robust.
Mm.
But the downside is that it really limits the number of stimuli that you can tell apart. And so we and animals in general, for example, like you can't tell the difference between two different sugars if they're matched for intensity. So like fructose, which is in your yeah, like sweet drink versus
Yeah.
table sugar, like it all tastes the same, right? Um, and that's because they all activate the same neurons. So that's kind of like the up and down side of this. So when it comes to salt, though, there had been a lot of debate over, well, there's a lot of data in general, actually, whether that's even true of any taste in mammals, whether this like separation of different modalities is really true. But this especially becomes complicated with salt because salt seems to activate not just one dedicated population of neurons. And so what we showed in that paper is that yes, actually, salt causes the activation of every taste neuron, not the activation, but changes the activity of basically every category of taste neurons. And so it causes most neurons to be activated, some neurons get inhibited, but we think that it's the combination of activating these different pathways that ultimately results in the fly's behavior to salt. And what makes this particularly appropriate for salt is that salt is unusual as a taste as well because the concentration of it determines whether it's attractive or aversive, right? So, like sugar at any concentration, despite what people might say, about something being too sweet or whatever, like sugar at any concentration is positive, and bitter compounds at any concentration are negative. Whereas salt at low concentrations, like on French fries or something like that, is really delicious. But then if you swallow seawater, it tastes disgusting, right? And so there's this flip in what we call the valence, which is like positive versus negative of salt taste depending on how concentrated it is. And so it makes sense that it activates a whole bunch of population or a whole bunch of different neurons because you have to, at lower concentrations, activate neurons that are attractive, and then at higher concentrations, activate neurons that are um, aversive.
Yeah, that's really fascinating. Actually, for listeners who don't know, could you talk about how you figure this out? So your research project process, sorry, for that 2018 study on salt.
Sure. So we typically take a number of different approaches in the lab, and we used all of those sorts of things in that paper. One of the big ones is to just look at the activity of neurons in response to different stimuli, and we do that using calcium imaging. And so to take a step back, like, when a neuron is active, when it's firing, many people, I think, know that neurons use electrical activity, right, to conduct their signal. And so they fire these things called action potentials. And what that means is basically like the electrical charge on the membrane of the neuron changes very rapidly. And so one of the things that happens in a neuron when you get those changes in the electrical activity is that this ion, calcium, actually uh, enters the neuron and also gets released from stores within the neuron. And so the calcium concentration inside a neuron will rise when it's active. And so we express these proteins that are sensitive to calcium concentration, and their fluorescence changes when the calcium concentration changes. And we express them in different populations of neurons and then measure those changes in fluorescence when the fly is tasting something. And that way we can measure the activity of those neurons in response to the tastes. So that's one thing we did, and we looked across different populations of neurons and applied different taste stimuli, mostly focusing on salt, to measure their activity. The other thing we did is to use some of the techniques available in flies to silence different neuron populations. And what that allows us to do is ask the question of like, what are those neurons necessary for? So if you give the fly, for example, a choice between, um, attractive concentration of salt and then water, a regular fly will eat the salt. And but what we can do then is go through and systematically silence different populations of neurons using these genetic techniques, and then ask, okay, when flies are lacking the function in this population of neurons, are they still attracted to the salt? And so we went through different populations and identified which neurons are required for attraction to different tastes. And then what we did was kind of the opposite experiment, which is to activate the neurons in the absence of the actual stimulus. And so you can ask the question like, okay, if I activate these neurons that I know are involved in detecting, let's say, high salt, is activation of these neurons alone enough to make the fly dislike something, right? And so we do that using this technique called optogenetics, which basically is the use of light to change the activity of neurons. And so if you express, for example, a channel that responds to, or a protein that responds to, let's say, red light in a population of neurons in the fly, like neurons detecting sweet, for example, and then you shine red light on the fly, in theory, it thinks that it's tasting something sweet, even though it's not. And so then we can measure the behavioral response of the fly when we activate different populations.
Wow, that's a very complex process, I think. I'm wondering how long did it take you to complete this study?
That particular paper, I think about 3 or 4 years, something like that. It was started by a master's student in the lab and then taken over by a PhD, by a postdoc when she came to the lab. And so together, I would say maybe 4 years, which is pretty typical in the field for how long papers take it. Anywhere from sort of 2 to 5 or beyond years.
Yeah. And while you were doing, or while the paper was being published or in the process of being published, and in the years afterwards, do you know how well the information you gathered from it translates to actual studies of the human brain and how it interacts with salts?
Yeah. So we do know some of the similarities that are present with humans. For example, in humans or in humans and mice, like okay, I'll state that a lot of what we know about taste is done in mice, right? So we can talk about humans that in the field, people tend to sort of loosely assume that what's happening in mice is also happening in humans, even though we know that there's many examples where that's not true. But okay, so if we kind of look in a general picture though of what's happening in mammals, um, one big similarity is that we found that flies use one population of neurons for attraction to salt, to like lower concentrations of salt, and then there's two populations of neurons, two different populations that both respond to higher concentrations of salt, and both of those will then cause the fly to avoid the salt, right? And so that's that's how it achieves this balance between being attracted to low concentrations and avoiding high concentrations by having these aversive pathways kick in as the concentration gets higher. And so the same is basically true in mammals. So believe it or not, we have populations of cells on our tongue that are specific to sodium. And so that's why we love sodium. And activation of those cells gives us that like delicious salty taste. But at higher concentrations, salt will actually activate two other pathways in on our tongues, which are sour and bitter. And so, in theory, if you put a bunch of salt water in your mouth, you are actually activating the same cells that respond to like, you know, uh, a lemon and also something bitter like Brussels sprouts or something like that, right? Like, yet it sort of creates this new sensation of high saltness. So that's one similarity, I guess, between flies and mammals.
Yeah, and if we actually go to sourness, so if we say lactic acid, I think believe it was your lab that found it was attractive
Right.
to flies. Yeah, so how does this differ from how humans, I think you're already on that topic, but humans perceive sour taste, and what might be the evolutionary reason for this difference in flies?
Yeah, so this is where we get into some complicated stuff. And we I alluded to how what we see in mice isn't always what we see in humans. And sour taste is actually one example where it's really unclear exactly what's going on with sour taste in mammals. So, acids, sour taste is a detection of acid, right? And so, there are certain cells on the tongue that are acid sensitive, and they have a particular acid receptor. And but the valence, right, that I
Mm,
talked about earlier, positive versus negative, is a little less clear in in anything, but especially in humans. And what that has to do with actually, and this is maybe getting into territory that's a little bit too esoteric for your audience, but
mm,
what happens is that let's say you drink something sour, it has a high acid content. It's activating sour cells on your tongue, but it's actually also activating pain neurons in your mouth. And so separating those two things is a challenge, and like the sort of relative contribution of those two things can get confusing. And so, like in humans, despite what I said about, like in the case of sweet, where I don't necessarily believe that things are too sweet,
mm,
there are like the concentration and the specific acid involved can make a difference in whether we are attracted to it or not. Um, okay, so that is also true in flies. And but in both cases, we don't really fully understand all the details of what's involved. So what we showed with lactic acid is that flies, for whatever reason, are just really attracted to lactic acid.
mm,
And there's lots of what we call carboxylic acids in the environment, right? So these are like organic acids like lactic acid or acetic acid, which is vinegar, right? Or other similar things. And flies like a lot of these, but they just love lactic acid. And we don't fully know why, but it might have to do with the fact that they like rotting things, and there's certain
mm.
species of bacteria that produce lactic acid when they undergo fermentation. And so maybe their food is higher in lactic acid, and it's become a positive cue for them. Um, right, but the confusing thing is then not to get into too much chemistry, but an organic acid like this, like a lactic acid, this carboxylic acid, it actually, like if you put it into solution, like in water, it has two things, right? We call it like the proton, which is the hydrogen ion, and then it's got the anion, which is lactate, right? And so What we think is that both of those chemical species actually influence the fly's response to lactic acid. And so there's a general sort of mild attraction to acidity, though not as long as it's not too much acid. And that's kind of similar to what I talked about with salt, right? Where like we like some, but not a lot. But then also, for some reason, the anion matters too, so the lactate. And we think that in flies, those two chemical species are actually activating different populations of receptors. So we have specific like protein receptors for protons and then other ones for the anions and like those that second family of proteins is important for differentiating between different anions, right? So they tells the fly whether it's lactic acid or acetic acid or malic acid or citric acid or
Mhm.
whatever.
Yeah, wow, that does sound very complex. But I
Yeah,
did
we're
think
still trying to figure it out, but anyway, yeah, go ahead.
I think the way you put it is really clear,
Okay,
for such a
thanks.
complex topic. Yeah. And moving on to nutrient sensing, I think we already touched up a little bit on that too, but
Sure.
your lab was among the first to show that nutritional sugars have taste-independent effects on feeding. So can you maybe explain the difference between taste and nutrient sensing and why both are necessary for a fly to make really good dietary choices?
Sure, it's a great question. So, what I've argued to this point in our discussion, I think, is that taste is actually, like you suggest, is kind of a nutrient sensor, right? So, like you use taste to detect different compounds in the environment, and those that reflects different nutrients. So you have taste receptors for, sugars, and that tells you that it's like got calories, right? And et cetera. So, what do we mean by nutrient sensing? Generally speaking, it's kind of like a broad category that is anything that's not taste. And so, if an animal ingests like a sugar solution or, a piece of cake or something like that, the theory is that yes, taste responses will tell the animal that it's sweet and whatever else it is, salty or whatever. And that will inform whether the animal wants to eat it or not. But at the same time, once it is ingested, there's also sensors in the body that will detect changes in nutrient levels entering the system that can then feed back to either increase or decrease um, the ingestion, right? And where it gets complicated is that the same signal can mean different things depending on the context, right? So like let's say I haven't eaten all day and then I there's a piece of cake there, right? And I start eating it. Obviously the sweet taste is going to be delicious to me, but then as I ingest it, to a certain point, you might think like, well, all those calories, like I'm calorie starved at this point, right? So like all those calories are actually a good thing, like I could die if I don't eat for too long. And so if my body is sensing the intake of calories, it should say, okay, like keep eating, right? Like we need this. But at the same time, at some point, that same detection of calories needs to tell the body, like, okay, enough. Like, you got to
Yeah.
stop eating this cake because, like, you're gonna be too full and gain too much weight and all these things, right? So it's the job of these nutrient sensors, in theory, to like mediate that balance. And we know in both uh, mammals, so again, rodents, right, and in flies, that they do have internal sensors that do just that. So if the fly's hungry or the a mouse is hungry, then it's like a positive signal, and if the animal is full, it's a negative signal. And there's different ways, at least in flies, that we think that signal is sort of like carried out.
Yeah. And actually, while you were talking, I kept imagining your research lab or your field in general as like I don't know, a fly. And there's so many directions you could continue to go ahead and explore on a already very well-built foundation. And so on that note, what's like the most exciting new direction in your research that you're hoping to continue to pursue?
Yeah, so I'm interested in several different things. But one thing that I'll talk about maybe briefly that I we haven't touched on today, is is the importance of taste in other insects and other animals. Okay, so like very naturally, our discussion today has focused on, well, what can we learn from fruit flies that'll tell us about our own brains? And that
Yeah.
it's an incredibly important question, and like that's a big part of a lot of what we've done over the years. But what I've started to become more interested in the last few years is also like what can we learn in fruit flies that will tell us about the taste of other insects? And how other insects taste and what they do in response to those tastes is actually an incredibly important health problem as well.
Mm.
So most notably, mosquitoes will taste you when they land on your skin and decide whether or not to bite you. And if they do bite you and you're in, you know, a lot of different places in the world, not here, but a lot of places, it might transmit a disease, right? And so mosquitoes are known as like the deadliest animal on Earth because even more so than humans, they kill more people every year through spreading malaria and other diseases. And so if we can understand how that process occurs in other insects like that are important, like mosquitoes or agricultural pests that are a big negative impact on the food supply, et cetera, it can actually have a effect on human health in a more sort of indirect way, in a sense, but maybe more sort of closely tied to the biology of fruit flies. And so we're interested in understanding or discovering receptors in flies and then their counterparts in mosquitoes and studying their function, and also doing the same thing with other agricultural species. Like there's another species of Drosophila actually called Drosophila suzukii that is a big agricultural pest because they lay their eggs on fruit that we might want to eat, whereas melanogaster, they lay their eggs on fruit that's already on the ground and rotting. So, and then it causes the destruction of a lot of fruit. So I've become more interested in sort of branching out into other insects as well to kind of probe at that angle of like application beyond the more general sort of like, it's important to understand the brain kind of thing.
Yeah, I think that's really cool. It's really interesting to use the melanogaster as a sort of like a blueprint for other insects, a more easily accessible and easily researchable. I'm not sure if that's a word, but um, sort of blueprint for other insects.
Definitely,
That's really cool. And yeah, I think research is a lot about translation, right? What can we use from this study to branch out and look at something else?
for sure.
That's and last question for today, but I guess for your research plan to branch out to other insects, is there like a dream device that if that could be developed in the next 10, 15 years, it would really significantly help your research?
Hmm. Device. Interesting. I have not. Hmm.
Yeah, it is a really broad question. It
It
requires you to
is,
think really
you
outside the
know,
box, I think.
it's an interesting question, yeah, because there's so much technology that's developed all the time.
Hmm.
And a lot of it, like, I don't have practical access to. So the question is not only just like, is there a theoretical thing that could be developed that would help, but also, what other things have already been developed that are helpful, but could be scaled into a way that makes them even more useful, to a broader population. And so, in the absence of an answer to the sort of in the first category, one of the things that's really sort of changed fly neuroscience recently is the connectome, right? So like this map of all the connections between all the different neurons in the fly brain. And so, you know, this is an enormous project that has taken many years, but it used to be that way for things like genome sequencing, right? Where people would take years and years to sequence the genome of a single animal, and now it can be done in a day, right? And so if something like the technology for studying connectomes could be scalable in a similar way, and we could then easily get connectome information from other species, for example, other species of insects or other larger species, etc. I think that would really, really accelerate everything a lot. So maybe that's something sort of that's, you know, more of an issue of scale rather than developing something new.
Yeah, I think that's a good one, I would say. And I think that's all the time we have for today. But thank you so much, Dr. Gordon. Your lab is, I think, it was the interesting approach to the melanogaster that really caught my eye. So I'm really glad that we got to dissect that today.
Sure. Thanks for taking the time, and thanks for coming up with great questions. It was really interesting to talk to you.
Yeah, thank you so much, Dr. Gordon. Have a good rest of the day.
Okay, you too. Bye.
Thank you.